Welcome learners for today's video class DNA fingerprinting technique. DNA fingerprinting, also known as DNA profiling or genetic fingerprinting, is a biotechnological technique used to identify individuals based on their unique genetic makeup. It analyzes specific regions of DNA that are highly variable, which is called polymorphic among individuals, making it a powerful tool for forensic investigations, paternity testing, and genetic studies. And this technique was developed by Sir Alex Jeffreys in 1984, who discovered that certain regions of human DNA contain repeating sequences that vary from person to person. DNA fingerprinting is based on the fact that 99.9% of human DNA is identical, while the remaining 0.1% contains variations, that is mostly in the non-coding regions, that can be used for identification. And the variable number tandem repeats and short tandem repeats are the key components which is analyzed in the DNA fingerprinting technique. Steps involved in DNA fingerprinting. The first one is DNA sample collection, that is DNA extraction. DNA is extracted from biological samples like blood, saliva, hair, semen, skin cells, or bones or tissue samples, followed by DNA extraction, which is done by chemical or enzymatic methods. Then DNA amplification, which is done by the polymerase chain reaction or PCR technique. If the sample contains very small amount of DNA, then the sample is amplified using the PCR to generate multiple copies for analysis. And this step is very crucial in forensic cases where DNA may be available in tiny amounts. Followed by DNA digestion, which is done by the restriction enzyme cutting technology. So in methods like restriction fragment length polymorphism, specific restriction enzymes are used to cut DNA at known sequences, and the resulting DNA fragments vary in length based on individual genetic differences. Followed by gel electrophoresis. The DNA fragments are then separated based on size using agarose gel electrophoresis or polyacrylamide gel electrophoresis. Smaller fragments travel faster, creating a distinct banding pattern. The next one is Southern blotting. If RFLP method is used, Southern blotting involves the DNA fragments which will be transferred to the nitrocellulose membrane or the nylon sheet, which is then allowed to hybridize with the radioactively or fluorescently labeled DNA probe that binds to specific repeat sequences. Followed by detection and analysis, the DNA pattern is visualized using autoradiography, X-ray film, or fluorescence detection. The resulting banding pattern, that is DNA fingerprint, is unique to each person and can be compared between samples. Applications of DNA fingerprinting. It is widely used in forensic science, paternity and maternity testing, medical diagnostics, wildlife and conservation biology, evolutionary and population genetics. And the advantages of DNA fingerprinting includes it is highly accurate and reliable, requires only a small amount of DNA sample, it can be automated for high throughput analysis, and it is used in various fields including forensic science, medicine, and anthropology. Limitations of DNA fingerprinting. Contamination can lead to inaccurate results. It requires specialized equipment and trained personnel. Ethical concerns regarding privacy and misuse of genetic data. Thank you all.